thank Links for this great initiative. I mean, I think creating this digital platform is really an excellent idea, especially at these times. And I'm particularly thinking about all our young scientists and the students who, you know, unfortunately don't have the opportunity to do experiments at these facilities or to even visit them. And so I think this is a very, very important initiative that we have now. So I'll be talking about the European XFEL, but before I start describing this facility, I want to say a few words about, you know, what is so special about X-ray free electron lasers and how they differ from, you know, the more familiar synchrotron radiation sources. And actually, you must know that XFEL radiation is very different from synchrotron radiation. First of all, as you can see here on the right, the pulse duration is much shorter. It is of the order of a few tens of femtoseconds with respect to synchrotrons, which is closer to 100 picoseconds. Then also the number of photons per pulse is orders of magnitude higher, as you can see here. The spatial distribution of the photons and also the energy spectrum is very inhomogeneous if you compare it to what you have at synchrotrons, which you can see here on the bottom. And finally, there are very strong pulse-to-pulse fluctuations, as you can see from this little movie. And the reason for these fluctuations is because these pulses are generated by a stochastic phenomena. And so this is the self-amplified spontaneous emission process, SASE. And I'll try to explain this with a little cartoon. So just like at synchrotrons, X-rays are produced by emission from accelerated electrons. So you have an electron bunch that is accelerated. These are relativistic electrons, and they go to very high energies, and then they enter into long periodic magnetic structures. Now here, I'd like to highlight two differences. First of all, in XFELs, typically, the bunches are very collimated, they're tightly collimated. That means the emittance is typically much smaller at XFELs than at synchrotrons. And the second difference is that these undulators are much longer. They're typically hundreds of meters long, whereas at synchrotrons, you have only a few meters long undulators. So this bunch comes in, and at the beginning, it starts emitting X-rays at every turn. So at the resonant wavelength. So it creates electron density fluctuations at this resonant wavelength. As the bunch progresses through this very long undulator, these electron density fluctuations are amplified. And at the end of the undulator, you have the formation of these micro bunches. And within each micro bunch, you have all the electrons that emit coherently. That means that the number of photons that are emitted goes as the square of the number of electrons in the bunch. And this is why you have these very sharp spikes in the spectrum. So as I said, a specificity of these facilities is this very short pulse length, which is approaching the femtosecond. And so this means that now, we can start looking at, ultra fast science, which has been up to now, the domain of optical lasers. Except now we have a wavelength which approaches the angstrom. So we are becoming sensitive to atomic positions. We have chemical selectivity because you can tune the energy of these photons to an absorption edge in a specific species. You also have bulk sensitivity because you have hard X-rays that have very large penetration depths. And so we're seeing that these facilities are really providing us completely new opportunities to study matter at time scales that are typical of electron dynamics and also phonon dynamics. Also, these very short pulses and extremely brilliant pulses are changing the way we do structural determination with X-rays nowadays. And this is summarized in this slide, which shows from 20 years ago where theoretical simulations showed that you could, have very, very high X-ray doses delivered in very short pulses, and you could obtain structural information before radiation damage actually destroys the sample. And this theoretical prediction has been experimentally verified. And today we are able to study extremely small crystals, radiation sensitive molecules and proteins, and we can do these studies at room temperature. And this means that we can start looking at the dynamics, you know, of these systems. Okay, so now I'm going to try a little bit these images here. Okay, I'm going to try and introduce this facility. So the European XFEL is an international user facility. It is funded by 12 shareholder countries, but mainly by Germany and Russia. The company, the European XFEL company, is in charge of the construction and operation of the X-ray laser, whereas DESY has constructed the accelerator and is now operating the accelerator. So we have a staff of 650, of which 250 are at DESY. The construction of the facility started in 2009, and the start of operation in the middle of 2017. We're offering both soft and hard X-rays, and we cater a pretty large user community, although not as large as the one that you might find at synchrotrons. So to give you a little bit an idea of where this facility is placed in, in the context, in the worldwide context, I'm showing here this map, which shows the operating XFELs worldwide. So in dark blue, you see the hard X-ray facilities, and in orange, the soft X-ray facilities. So we'll be focusing now just on the hard X-ray facilities. And LCLS was the first one to become operational in 2009 in Stanford. And then in the following decade, there were 4 more facilities opening to users. We have Sakura in 2011. Then we have PALFEL and the European XFEL in 2017, and finally Swissfel in 2018. So among these facilities, the European XFEL was the first hard X-ray free electron laser to be powered by a superconducting accelerator. And this allows it to reach, very high repetition rates. And at the European XFEL, we work with megahertz repetition rates. So LCLS is also being upgraded, and it is constructing a superconducting accelerator, and this will enter operation very soon, although at the beginning it will be limited to the soft X-ray regime. So this is a layout of the facility. It is located close to Hamburg in Germany. So here on the bottom right, you have the city of Hamburg. And then you see here in this area, this is the DESY campus. And you can see here the ring, Petra 3 ring. Now, our injector is underground in the campus, and the accelerator is constructed inside an underground tunnel. This tunnel is about 2 kilometers in length, and you can see here a picture, a photograph of the superconducting cavities. At the end of this accelerator, electrons reach a maximum energy of 17.5 GeV, and then they are injected into different undulator photon tunnels. So here they interact with the undulators, and they produce X-rays that are then used in the experimental hall that is located underneath this administration hall. So I'm just showing you a few pictures, historical pictures. This is the signing of the International Convention in Hamburg Town Hall. This was in November 2009. You see the representatives of the different shareholders. These are photographs from the tunnel completion ceremony in June 2012, with a speech given by Massimo Pascarelli, who was the former chair of the management board at the time. And here are pictures from the inauguration ceremony and start of year operation in September 2017. And you can see here this wonderful picture of this laser beam, which is sent out from the Elbphilharmonie in the center of Hamburg in the direction of our facility. Okay, so I want to focus here on the beam distribution and the instruments. So as I said, the electrons are accelerated to a maximum energy of 17.5, and then they enter into different photon tunnels. So at the moment, we have 3 undulators, 3 photon sources. 2 of them are for hard X-rays, SASE 1 and SASE 2. And this third one is dedicated to soft X-rays. So you see each of these undulators feeds two different instruments. So at the moment we run with 6 instruments, 4 of which are hard X-rays and 2 of which are soft X-rays. And 3 of them at the time can work simultaneously. We typically work on 12-hour basis, but this may change in the future. So here are, this is a table which summarizes the key parameters of this facility. So I already talked about the electron energy, maximum energy of 17.5 GeV for the electrons, and we reach a maximum photon energy of about 25 kV. I'd like to focus a little bit on the delivery pattern, because this is very specific to our facility. So it's based on the fact that we have a radio frequency that runs at 10 Hertz. So this means that we produce macro pulses every 100 milliseconds. And within these macro pulses, which we also call trains of pulses, we can fit up to 2,700 pulses. So at a maximum spacing of 200 nanoseconds, which is 4.5 megahertz. Each of these trains, as you can see here, can feed the 3 undulators. So each of the trains, so the 3 instruments can run actually at the same time. So all the 6 instruments are now in user operation since the end of 2019. I'm gonna present them to you here. So on the left, here we have the SASE-1 hard X-ray instruments. These were the first to become operational in September 2017, so at the inauguration of the facility. So on the top here, we have the single particle and biomolecule instrument that also focuses on serial femtosecond crystallography, led by Adrian Mancuso. So this instrument is really catering the biology community, but not only, mainly the biology community. Here on the bottom, we have the femtosecond X-ray experiment instrument led by Chris Milne, and this is really catering the chemical dynamics community. Here in the center, we have the 2 soft X-ray instruments that started operation at the end of 2018. So on the top, we have the spectroscopy and coherent scattering instrument led by Andreas Schertz. And on the bottom, we have the small quantum system instrument led by Michael Meyer. And finally, here on the right, we have the SASE-2 hard X-ray instruments. So these are the last to become operational in the middle of 2019. On the top, we have the material imaging and dynamics instrument led by Anders Madsen, and here the high energy density instrument led by Ulf Zastrow. So this is a nice collage of photographs taken in 2019. So you see a lot of happy and very crowded instrument control hutches. And as you know very well, the situation has changed drastically at the beginning of 2020. So before I start talking about the science, I just want to mention here, the impact of this pandemic on the operation of our facility, which has been pretty drastic. So the virus started spreading in Hamburg at the beginning of March, and we entered a reduced operation mode in the middle of March, where we switched off our accelerator and we stopped all experiments. And then we gradually restarted the facility in May and June, and in the second half of the year, we could carry out a limited number of experiments, a very small number of experiments. Then we had planned a normal shutdown, and we had planned a normal restart in 21. However, the situation degraded very quickly before Christmas, and we were forced to have a much longer shutdown than we had foreseen. And the restart was also delayed and was much longer. And therefore, we still are far from a normal operation. In total, we had 3 COVID-19 infections within our staff, but none of them inside the facility. We had very few users, as I said, a total of 16 that were allowed on site in 2020. But we are actively involved in COVID-19 research, and we have now 3 experiments that are coming up. So just to give you some numbers, out of the total 72 experiments that had been either scheduled or shortlisted for 2020 from Run 5 and Run 6, only 23 could actually be carried out in 2020. Many of them had been postponed to the first half of this year, but even then we could not schedule them, and so they have been rescheduled to the second half of this year. Now the coming runs, Run 7 is the one that was called just before Christmas, and the proposals that will be selected from this run will be scheduled in the first half of 2022, and we hope to have finally a regular call for proposals in November 2021. Okay, so now I want to focus a little bit on the science. So I told you about the facility. I ran through quickly the instruments, and now I want to talk about results. So as you can see here, we start to see publications coming out from all the instruments. I've picked a few of these as examples, and I will very briefly go through them just to give you a little bit of a flavor of the kind of science that we're doing at the European XFEL. So the first example is devoted to megahertz serial femtosecond crystallography. And I'd want to underline here that the first year, actually the first 2 years, we really focused on trying to see how we could exploit this megahertz repetition rate, because this is a real challenge. I mean, every aspect of the experiment has to be compatible with megahertz repetition rate. And this goes from sample preparation to sample delivery to data acquisition and etc. In particular, for the serial femtosecond crystallography, the sample delivery was a real challenge. You see, if the jet is too thick or too slow, as shown in this image, then it will not recover in time for the next pulse. And so they had to develop special delivery methods with jets that were much faster and much thinner than previously. And what I'm showing you here are first results from one of the first papers where they were able to solve the structure of an unknown compound, which was in this case involved in antibiotic resistance. So this really demonstrated that we are able to fully exploit today this megahertz repetition rate. This other example is, this is the first work that showed that we could actually follow the structural changes in a protein throughout a biological process using pump and probe methods. So what you see here are different, let's say this is a time series of serial femtosecond crystallography data. So these are electron density patterns and differences, as you can see here, during the photocycle of the photoactive yellow protein. So the data here in these three images was taken at the European XFEL. And it perfectly, let's say, links previously obtained data from, this is from LCLS, to the data that can be obtained on the timescales of 100 picoseconds, which is typical of what you can do at synchrotrons. So you can link the data from LCLS to APS doing with a sort of like a movie. And so this was the first example showing that these molecular movies is something that now is becoming possible. Okay, another important application, I mean, for these facilities, these X-ray free electron lasers, was to determine the structure of ideally a single biomolecule at atomic resolution. So this is a big challenge, and at the moment we're still not there yet, although worldwide there's a lot of efforts going in this direction. The technique used is single particle imaging, where you practically collect a coherent diffraction pattern from individual particles that are dropped into the beam. Now the challenge here is to be able to have for each of these patterns a sufficiently low background so that each pattern has statistically relevant information, so that then you can use, the collection of these patterns, and you can apply reconstruction algorithms and reconstruct the three-dimensional image of your particle. Now this summer, last summer in 2020, we had a first paper that was published, which demonstrated that thanks to the megahertz repetition rate, we have so much data that this really allows to overcome many of these challenges. So this work was on a collection of a population of tiny gold nanoparticles of different sizes and different shapes. And the algorithms developed here were able to reconstruct this full population just using statistical methods. And as you can see here, also with pretty good spatial resolution. So this really tells us that these megahertz facilities have, will open a lot of opportunities in this kind of applications. The megahertz repetition rate was also used to do high-speed imaging with high spatial resolution. So what was done here was a movie of a very fast process. So here we can start looking at processes with speeds up to several kilometers per second. What is interesting is that these methods will then be, extended to to obtain three-dimensional information. So to do movies where you have full three-dimensional information using projection tomography. And these will be very, very interesting in the future for industrial applications. So this first experiment looked at a liquid-filled glass capillary that was hit by a laser and it explodes. And what you can see here is you can see the shape, the orientations, and the velocities of all these microscopic, micron-sized glass and water particles. And the contrast and the resolution that you get on these images is very, very complementary to what can be done today at synchrotrons. Okay. This is experiments. This experiment was devoted to studying heme proteins. So these are very important proteins because they are responsible for many cellular processes in our body. They also carry small molecules such as oxygen in our blood. Now, for example, it is known in myoglobin that when these small molecules bind and when they are released, this heme ring, you see that the protein is based on a heme ring with an iron atom in the middle. So this heme ring goes from a relaxed state, a planar state, to a tense domed state. It just bends and the iron pops out of the plane. And up to now, it was not, it's not really clear why this occurs. And also what is not clear is, the intermediate states and, what causes this change in spin and in structure. And also what is not known is whether this is something which is specific to myoglobin or whether it is a universal feature that can be extended to all heme proteins of this kind. And so this group worked on cytochrome C. They artificially cause the release of the small molecule using a laser pulse, and then they follow the local structural changes around iron and the changes in the spin state of iron using femtosecond resolved X-ray absorption spectroscopy. This was done at XFEL, and femtosecond resolved X-ray emission spectroscopy, and this data was taken at the European XFEL. And what they see is that in cytochrome C, you have a very similar change in the local structure, which tends to confirm that this is a universal feature of these heme proteins. This doming effect. And they were also able to, identify a whole chain of spin changes that induce this structural transition. Okay, this is one example from the soft X-ray instruments. So the group was studying magnetic Skermions. So these are magnetic topological quasi-particles that can be very, very small. As you can see here, this is an image showing they can reach diameters of a few nanometers. So these Skermions are becoming now a hot topic because of the potential that they have in development of faster and more efficient data storage devices. So what the group did here was they created Skermions using an optical laser pulse. And they followed the nucleation and the annihilation of these particles using ultra-fast wide-angle scattering and small-angle scattering. And the whole process lasts a few hundreds of picoseconds. So they start from a uniform magnetic phase, so magnetic multilayer. They hit it with a laser. This phase is heated up into a highly fluctuating spin state. And then when this cools down, you see the nucleation of the magnetic Skermions. And what they understood is that the presence of this high temperature, highly fluctuating spin phase is key in overcoming the energy barrier between the two phases. So when the system cools down, this energy barrier is sort of protecting these newly nucleated Skermions, and it keeps them stable against, temperature fluctuations. And this is really important for these devices. Okay, this is our first science paper, which came out a few months ago. This is work from the other soft X-ray instrument, Small Quantum Systems, SQS. It describes a new method called photon recoil imaging, which is able to, let's say, visualize very rare nonlinear processes, which typically you don't see because they are, let's say, covered by the much stronger linear processes. So I'm going to try and explain what they did. So here they worked on, they had a collimated atomic beam of neon atoms coming in, and they make these neon atoms interact with an X-ray pulse, okay, coming down in this direction. And then they place a position sensitive detector just behind the interaction region. And this position sensitive detector is detecting the position of these neutral neon atoms that just continue to, to propagate after the interaction. And these neutral neon atoms are the product of the interaction between the X-rays and the neons, which is shown here on this little schematic drawing on the top. So the X-rays have just the right energy to excite the 1s electron of neon into this, to the 3p state. And then you see that the neutral neons that are detected here can be obtained in two ways. Either the excited neon de-excites itself through normal radiative de-excitation by emitting spontaneously an X-ray. So this is the normal fluorescence that we know. And this photon, which is this blue wiggles, goes isotropically everywhere, which means that this neutral neon atom will get a little kick in all directions when this photon leaves. And so it will end up here in a big area. And this is shown here on this image here, a large area. This is where these neutral neons end up. However, there are some of these neutral neons that go through a different path, and this is this nonlinear stimulated X-ray Raman scattering process. So this de-excitation is stimulated by a second photon, which is absorbed at the same time as the first one, slightly different energy, but exactly at the same time, and they're propagating in the same direction. And therefore, in this case, there is no kick, there is no recoil. And therefore, these neon atoms are just, let's say, projecting this interaction region on the detector. And so you see this little segment here. So this is a very nice, elegant way of identifying these very rare processes. And in the future, these methods will open up a lot of opportunities when we will be able to, fully control photons of different energies. And we are very close to that. We're working on that. This is an example from the high energy density instrument. So here we're focusing on producing and probing matter at very high pressures and temperatures. These states can be obtained, for example, using high power lasers and through dynamic compression. And they are typically transient states in the sense that they maintain these extreme conditions of pressure and temperature only for a very short period of time, typically from femtoseconds to maybe hundreds of picoseconds. And that's it. So HED is trying to develop diagnostics to be able, for example, to measure the temperature in these extreme states of matter. And the method shown here is based on inelastic X-ray scattering, high resolution, so 100 milli-electron volts resolved inelastic X-ray scattering, where so what they did here is they used a single crystal and they measured the energy change of the incident photons when they interact with phonons. So this was a solid. So they were looking at interactions with phonons, but it could be liquid, and then you could look at interactions with acoustic waves, for example. So the diamond was measured at ambient temperature, and it was heated, resistively heated to 500 Kelvin. And you can see here the differences in these inelastic scattering spectra with the Stokes and anti-Stokes peaks, which have very different ratio. And from these, you get directly the bulk temperature of your sample. So this is something that will be certainly applied once we will be able to have these lasers in operation. We're not there yet. Okay, this is my last example, which shows that these facilities also are opening opportunities in following very fast dynamic processes using high resolution imaging. So looking at the nanoscale dynamics of these systems. So what they did here is they used near field propagation imaging to observe the explosion of a microfluidic water jet caused by a laser pump. And they followed the dynamics by using single pulses delayed, at different delay times with respect to the pump. Now, the problem here, the main challenge here, is that you're limited by the intrinsic fluctuations of the SASI beam, because every single X-ray pulse is different, has a different intensity and different spatial distribution, etc. And so this group developed a process by which you could correct for these instabilities. And the images that I show here on the bottom, so this is a sequence, a time sequence during the explosion of one of these jets. So from 4 nanoseconds to 30 nanoseconds after the pump. And you can see here a lot of detail because it's no longer blurred by the instabilities. You can see how this jet is broken up, how the shock waves propagate along the axis of the jet. And these images can give information on density changes and pressure changes. And this is not available by optical methods. So these are very, very complementary methods. Okay, so I would like to underline that all this would not be possible without, obviously, a very strong collaboration between the facility and the user community. And here I would like to mention the role of user consortia. So these are groups of institutes, mainly from shareholder countries, but not only, that get together and decide to develop or to fund big projects like a full experimental station or a lab. And once these instrumentations are commissioned and become operational, they are then provided to the full user community, to be used by the full user community through the normal peer review process. So just to give you some examples, we have here the SFX consortium that funded a serial femtosecond crystallography chamber station on SPB. We have the HIBEF, the Helmets International Beamline for Extreme Fields user consortium that financed, among many other things, the high intensity laser and the high energy laser on the high energy density instrument. We have here below a full biolab. So this is an integrated biology infrastructure and life science facility, the XBI labs. So these are really big investments. And there's many more that I don't have time to go through. Now, one thing is that we have realized, on the other hand, that our user community is not very, let's say, homogeneously distributed among our shareholder countries. And so now we are focusing in the coming years in trying to increase, the diversity in the way this facility is used. So we would like to increase the scientific use by the smaller shareholders and by underrepresented shareholders. So we're trying to, make our experiments simpler. We're going to be working on, making standard configurations, offering standard configurations that are more user-friendly and that can be used by smaller groups. We will be investing in training and supporting new users. We want to encourage scientific collaborations with our staff. So we've already started a program of shared PhD students with universities from other countries. We're also encouraging new users and inexperienced users to match up or to work together with what we call super users. These are groups that systematically are successful in getting beam time. And finally, we are going to be implementing new access schemes, so to have more flexible access to beam time. For example, with long-term proposals, if you want to have a long-term development that requires more than one beam time. And also we'll be launching topical calls for proposals. We may have a call maybe on water in the upcoming months. So I have gone through everything I wanted to say. I just want to conclude by saying that I've showed you some first scientific results, but we still have many, many challenges ahead of us. The community is still quite small. I mean, if you compare it to synchrotrons. And so, as I said, we must make these experiments simpler to welcome, a larger community. Another challenge is that we are offering both soft and hard X-rays, and we have only one accelerator. And sometimes this leads to compromises, and we need to learn how to deal with these. And finally, we're still struggling with detectors. I did not have much time to go into technical details, but we are being submerged by data. And just to give you a little flavor of what this really means, you can see here the evolution of the raw data generated by the European XFEL instruments as a function of time from the start of operation. And you very clearly see the periods where we stopped operating. And now here on the right, you see the amount of data in petabytes per year. So raw data per year. And believe it or not, in 2020, which I mean, I showed you how calm that year was in terms of experiments. We did very few experiments. We still managed to, collect 10 petabytes of data in 2020. So this is really scary in the sense that at the moment, we still don't even have megahertz capable detectors on all the instruments. So we expect this to diverge in the coming years. So we are giving this problem a lot of attention at the moment. So an outlook, European XFEL is still a young facility. I believe that the coupling of hard X-rays to megahertz repetition rate, which is really something completely new, is opening new perspectives for science, and I gave you some examples. But a lot of the potential of this facility is still to be exploited. We will be going more in the direction of nonlinear phenomena with these very, very intense beams. We may be attempting wave mixing techniques using hard X-rays in the future. We want to better couple ultra-fast sources to our X-rays. We're also developing special modes. I did not have time to talk about this, like self-seeding, two colors. And finally, we certainly will go towards harder X-rays, and this is something that we hope also to have an exchange with the user community on this. And to do this, we are now organizing a series of workshops in the coming years. So for some of these, so this one has just taken place, but some of these, you see the dates here. We hope to see many potential users at these workshops. We really need input from the community on what are the future directions. Where would you like to see us go? And yeah, as I said, this interaction with the users is very, very important for us. So having said this, I would like to thank you for your attention, and I would be happy to take any question you might have.
Thank you very much, Sakura. It was a really great presentation, and I'm sure the audience agree with me. So we have a few questions that I can read on behalf of our audience. So we can start with the last question. Which one of the workshops are targeted at new users who don't know where to start. Like this is the last slide probably that you showed us. So one of the questions is really, which one of those workshops now you suggested is targeted for those that don't know anything, like where to start when?
Okay, good question. So actually, in these workshops, we would like to get a lot of people who are not familiar with XFELs. We really want to go beyond our little family of the usual ones. This is really the aim. So I would say that all of them are aimed at attracting this new community. And we have really made it a point not to focus on too technical things. We will not be talking about, you know, technical things of the accelerator or of the sources. We will talk about science. What kind of science do you want to do? This is the only way we think we can attract you, because if you are not a specialist of XFELs, you will not be interested in hearing about, you know, all these special sources or whatever. So we're going to keep that separate. So that's the aim. And then we will also be organizing more restricted sessions that are maybe more focused on one application or one instrument. So those are not listed there. These are a separate kind of meetings that we're also working on. And if you're interested in a particular scientific area that you already know you can do with the existing facility, you just contact us. You have the names of the scientists that I put there. So their emails is just their name, and then you add at xfel.eu. So it's very easy to find.
That's great. So now we have another question, which is a little bit more specific, and it asks, so with the challenging condition of the megahertz pulses and bunch patterns, what is the unique scientific aspect that the European XFEL feels that, for example, the LCLS2 cannot? So that's a comparison between another facility for the audience.
Yeah.
So
yeah. Let's say that there are areas that we think are uniquely, let's say, suited to this sort of burst mode.
Mm-hmm.
First of all, when you're using this burst mode, which is very specific to XFEL, I agree, you can concentrate more energy per pulse and have, of course, higher repetition rate, but only for a limited amount of time, only for the train. So this is very good when you're looking, for example, at movies, taking movies, for example, in microscopy, to look at a process which is short in time, like lasts less than a millisecond, but you want to have, many, many images, because we can take images every 200 nanoseconds, for example. For each of those images, since this is an irreversible process, you just have, they're all different. Things break differently. They don't break in the same way. So you need to have enough pulses. You need to have enough, photons in that pulse. So having a lot of energy or a lot of photons per pulse and being able to have a sequence, of very fast pulses every 200 nanoseconds will allow you to see these kind of processes. This is also something that we're also using a lot for high energy density science, looking at phase transitions triggered by the beam itself or by whatever lasers in the diamond anvil cell. So we're using the megahertz repetition rate to see the evolution of these phase transitions. These are reactions. So again, they happen and they're not exactly reproducible. So you want to have enough pulses in that one film to be able to see what's going on. So these kind of things.
That's incredible. Another question, is there research at the European XFEL related to the development of optical elements that can be applied with such intense beam?
Yeah, I mean, I think this is a worldwide challenge, and I think all facilities are working on this, and I believe that there is a lot of collaboration in this field between the optics groups in the different facilities.
So clearly there is an attempt to work in that direction too.
Yes, yeah. I mean, I know that there are platforms where they exchange information on metrology, on cooling, on how to make these optics more resistant to such high and intense beams.
That's great. And then we have other more general questions maybe. So what's the format of a normal experiment bracket? So it's several days or 24 hours, like in a synchrotron. So that's a curiosity question.
Yeah. Well, it's difficult to answer. I mean, today we don't have a standard experiment. Today at XFEL, unfortunately, we're not still in a situation where, you know, we are ready to run from minute one of the experiment. So there's still things that have to be tuned and tweaked. It's a young facility. This is a normal phase. There will certainly be experiments that will be over in a few hours, or maybe even faster, because we have a very high repetition rate. So you come in, you put your sample, and maybe after half an hour, you have all the data you need. I'm thinking about serial femtosecond crystallography and this kind of experiments where you have you can relatively standardize the setup, which could be suitable for a number of different samples. But then you will always have experiments that will require maybe 2 weeks of beam time. I mean, which are very, very time consuming, because it's just the alignment with the beam and the testing of the methods and things that are complicated when you have, you know, lasers and many other systems to synchronize. So we want to go in a direction where we offer both. We don't want to focus on one or the other. We want to be open and flexible, but we think that we will need both kind of experiments.
Okay, so about testing, so what sort of experiments have been run in terms of in situ testing? I suppose like before, like the real beam time, or.
So I don't know what the question really wants to get to, but we are going to open the possibility for users to come and screen their samples, for example, to test their samples. We're setting up a chamber in, for example, in the labs, in the sample preparation labs, where you have the injection systems and you have the same exact chamber that is on the instrument, so that you can come and you can test the delivery without wasting any beam time. So
that's great.
If the injector gets clogged, the nozzle gets clogged or whatever, so that you can really make sure that everything is perfect before you even start going to the instrument. This is something that we're putting in place from the next run.
Wow, that's really good from a user perspective, I think. And we have another question. So what are the future perspectives? So what techniques can we expect to see coming up in the coming years? But I think maybe it was also a little bit included in your presentation already.
Yeah, well, I didn't have time to talk about everything. I would have loved to talk about many other things that we're testing now, but I wanted to talk only about published results. Because you can imagine, in a young facility, we have many things that are going on which are not yet published. So, for example, I think that there's a lot of potential in X-ray photocorrelation spectroscopy to look at fluctuation dynamics in systems at equilibrium and out of equilibrium. And here again, the megahertz repetition rate is very valuable, because what you do is you have a system, let's say at equilibrium or whatever, and then you come in with a pulse, the first pulse or the first pulses of the train, and then you do something to the system. You say you heat it or something happens. And then the following pulses that come every 200 nanoseconds or so probe the dynamics on this time scale. And this is a very difficult time scale to access. This is the microsecond or the sub-microsecond time scale. And there's a lot of dynamics in soft matter and biology that happens on this time scale. So you take an image, let's say a coherent diffraction pattern with the first pulse, and then you take another one with the second, another one with the third, another one with the fourth, and you can do differential XPCS analysis of those images. If nothing changes in the sample, the images will all be identical, right?
Yeah.
But if something is changing in the sample, the images will be different, and you will be able to extract the changes in the dynamics from those images. So that's the thing.
That's crazy interesting. So of course the audience, Ryan, thank you very much for the amazing talk, very good presentation. And
there's one related question with the normal format of the experiment. You said few experiment can finish in maybe in a half a day or something. So how can one schedule or arrange those sort of experiment? What is the procedure?
Yeah, very good. So this is new, right? At the moment, we are very, very traditional. I mean, we only have one kind of access scheme. You put your proposal and you get beam time every 6 months. So this is the way we've been working up to now. But starting from the next run, we want to be more imaginative and you know, provide more flexible access schemes. And one of the access schemes is to use standard configurations, for example, where you will adapt your experiment to the configuration. And so you come in with your sample and you don't have to touch anything. That's the very short experiment that I was talking to you about before. So in this case, you write a proposal and we will guide you saying, you know, on the website, we will have a page which describes the standard configuration. And we will tell you, if you are able to use that standard configuration, you will have much higher chances of getting beam time.
wow.
You can see many more proposals. And we plan to have 2 different piles for the peer review panel. One pile for the standard configuration and one pile for everything else. And clearly, I mean, they will be peer reviewed. So if it's not interesting, you know, the proposals will not be accepted. But those that will be able to be done more rapidly because they don't need additional setting up. And so that's the way we're going to try and do it, starting from the next call.